Hello, this is Alejandro Montijo, and this is a video presentation for Bio 282 Lab at Arizona State University on the comparison between the primary literature, Scaffolding Proteins Guide the Evolution of Algal Light Harvesting Antennas, and its news article counterpart, Mystery of Photosynthetic Algae, Evolution Finally Solved. The news article details how cryptophytes have a very unique antennal protein structure when compared to its red algae or cyanobacteria counterparts, and this structure was unknown and is what baffled scientists for a very long time because when looking at it from an mRNA sequence standpoint, which is very common for scientists, it was vastly different, as stated here in this first key point, and therefore caused it to be unknown. However, the hypothesis or the theory of how cryptophytes managed to have this unique protein structure was thought to have been endosymbiosis, which is basically just where, in this case, an algae consumes another algae, but rather than digesting that algae, it keeps it alive, but it can harvest these abilities, and this was thought to have possibly been the cause of, or where the crypto fight managed to have received this unique structure was possibly from a different algae rather than red algae or cyanobacteria, as stated here in this second point. Furthermore, how this theory was confirmed, or how they found out that this cyanobacteria did in fact get this structure from red algae, was actually due to 3D modeling. So rather than mRNA sequences, they just looked at it with 3D models, and they managed to match these structures, and they can see similarities between them, as stated in this last point. Obviously, compared to a news article, primary literature is very detailed, especially on this first point, where cryptophytes were in fact known to have been receiving these abilities, just like their plastids, which is this photosynthetic organelle itself, was in fact from red algae. However, again, the antennal structure was just so different to red algae that scientists just didn't know where it received it. However, like the second point on the previous slide, the cryptophytes did in fact manage to get this structure from endosymbiosis. But the fact that it was from red algae is what also baffled scientists because, again, when looking at it from an mRNA sequence standpoint, it's just so different when compared to red algae. However, this was further explained as this happened due to fitness and endosymbiosis, because over time, after primary and secondary endosymbiosis, cryptophytes just further changed the protein structure, and through fitness, it just eliminated all those that weren't as good as what cryptophytes have now, and therefore is why or how cryptophytes managed to receive this structure from red algae, which will further be explained in the next slide. How cryptophytes actually obtain their antennal protein structure. So as you can see below my cursor, this is the ancestral red algae. And then these PBS linker families are just different variations of red algae. But as you can see here on LN6 below my cursor, these subunits, or this protein structure, further, this specific one at least, after primary endosymbiosis, as stated here, gene duplication or recombination, loss of the nucleus, it further changed these domains or these subunits. However, at this point, it was probably still similar in their mRNA sequences when compared to red algae. And then just after secondary endosymbiosis and after dimerization, which is just the combination or when something conjoins, it just further changed the mRNA sequence, which is what baffled scientists, and they were confused on why it was so different. But when looking at it like Professor Kermi, it's obviously seen that all these structures are very much so quite similar, and that is exactly how it was found that these structures are actually, in fact, the same, in a sense. But further detailing on how the news article did, these key points or these major points, obviously when compared to the primary literature, it's going to be a lot more generalized for the average reader. However, through and through, the news article did the primary literature justice. It is missing a lot of information, but again, the primary literature is just so detailed and very wordy with a lot of biological information that probably your average reader wouldn't understand. But all in all, these points are very thorough, especially when looked at in the primary literature. They're all prevalent, and therefore you can conclude that these news article, or this specific news article in this case, is in fact accurate. It's accurate because all these key points, they're all thorough, they're all prevalent throughout the primary literature. This is my works cited page, the first one being the primary literature, the second one being the news article, and then the last one being just the title screen's background. This has been Alejandro Montijo, and that concludes my video presentation for Bio 82 Lab. Thank you for watching.